A very good morning to one and all present here. Our esteemed guest, Professor Arijit Mukhopadhyay, Professor in Precision Health, School of Science, Engineering and Environment, University of Salford, UK, Head of Department, Biotechnology, Professor Yasha Hasija, Convener of the event, Dr. Navneeta Bharadwaj, all faculty members, staff, and students, we welcome you to the 4th lecture series in biotechnology organized by the Department of Biotechnology, Delhi Technological University. Now I request all the dignitaries on the dais to please proceed for the lamp lighting. Now, as we embark on this journey together, I request the Head of Department, Biotechnology, Professor Yasha Hasija, to please welcome our esteemed guest, Professor Arijit Mukhopadhyay, with a sapling. I request Professor Yasha Hasija to please deliver the welcome address.
Good morning, everyone, and welcome once again to lecture series in biotechnology. This, as we all know, is an initiative by our Honorable Vice Chancellor, sir, wherein we invite the dignitaries from India and abroad who have established themselves over years and share, as in VC sir's language, they share their experience of 20 years in, let's say, 1 hour. So this is, the best opportunity for you to grasp. And today we have a very esteemed speaker amongst us, Professor Arijit Mukhopadhyay. He is a dear friend of mine since my PhD days. So when I was doing my PhD, he was a postdoc, and you will see why we have him as a speaker today, because he'll speak for himself. So on these words, I welcome him again. And I also thank Dr. Navneeta for coordinating the event and all the faculty members and students for being present here.
Now, I would like to introduce our esteemed speaker, Professor Arijit Mukhopadhyay. Sir is a well-established leader in the field of genetic biomarker discovery and precision health, sustaining an excellent publication record. He specializes in research on disease genetics and genomics and have experience for nearly 2 decades working across multiple countries and continents. His research focus is early diagnosis of diseases using molecular genetic biomarkers for precise health management. He serves the University Senate for the Academic Ethics and Research Integrity Subcommittee and also as Staff Wellbeing Lead for the School of Science, Engineering and Environment, and also contribute as a part of the Mental Health and Wellbeing Working Group for the University. I request Sir to please deliver his lecture.
Hi, thank you Yasha. As Yasha says, I know her, that was 18 years ago. I was just counting in my head. I came as a fresh postdoc turned scientist. I did not know what that meant. I did not have a chair to sit, nobody in my lab. And she was one of the senior PhD students. So I felt more like a PhD student than a scientist those days. So kind of, we interacted with her and the whole batch. So a lot of memories. Thank you for bringing them back. Thanks for the introduction. It's very unnerving to listen about yourself from someone else. But thank you for that. I think it sets the stage. That's the title for the talk. It's going to be a little bit different. Of course, you are all, most of you I think are science PhD students, biotechnology PhD students. So there will be some science, but also beyond, as the title says, that I'll try to tell you a story rather than showing you a lot of data. So I'll tell you a story and tell you how the story has changed things that the data didn't. And that's what's going to be. So it's going to be on glaucoma. look at glaucoma as an example, meaning that what I'm showing you for glaucoma can be extrapolated to many other diseases, right? It's just a test case, it's a case study that this is something can be done for other diseases as well. So I am University of Salford, which is in Manchester in the UK. If people in the audience follow cricket or football, then you would know Old Trafford Stadium. So I live in there, not in the stadium, but I live in Trafford. So that's the context for you geographically where I am. And I never miss a chance to show off this lovely Victorian building. My office is at that building. So I'm lucky enough to sit in that building. My research lab is somewhere else, but just to give you the context where I'm coming from. So what happens in glaucoma? So let's say this is a visual that you are seeing, and you probably are seeing the whole screen. So you are using your peripheral vision as well as your central vision. If you're looking straight ahead, you can see a lot in the side. That's the normal sight. And when people have glaucoma, that peripheral vision reduces, and it goes on reducing. And this stage is called extreme glaucoma, where about 70% of the vision is lost. And then after this, the next stage is complete blindness. Okay, this is what happens. And what makes it more dangerous and interesting is that this process, from the beginning to up to this point, there's no symptom. So the person doesn't realize they're losing vision. There's no pain, nothing. Only they see that they have to turn their head more to see the sides. That's how they figure out. Most of the glaucoma gets detected accidentally when people go to check for cataract. Right? That's how it's detected. So that makes it more dangerous because it's irreversible. So put that in context with cataract, for example. Cataract is much more in number, if you look at number of patients, but that is reversible. You do a surgery, you get the vision back, in most cases. Right? Glaucoma, whatever you lose, is irreversible. You don't get it back. Okay? So that makes it more dangerous for a patient, interesting from a science point of view. Glaucoma has many subtypes, as many other conditions have many subtypes. It's a fairly common disease. The overall spectrum is fairly common disease. How common? So it affects around 100 million people globally. Right? It doesn't mean that all the 100 million are blind. They all have the disease. Some of them are blind, all of them are not blind, because it's a progressive condition. And these are all the subtypes. From a genetic point of view, because that's what I come in, the primary subtypes are predominantly genetic. Right? Not 100%, but predominantly genetic. Secondary subtypes are usually not genetic. What is a secondary subtype? For example, you get hit in your eye by a ball or something, and as a complication, you may develop glaucoma. Right? That's a secondary subtype. Has nothing to do with genetics. Primary subtypes are primarily genetic, and I will talk about a particular subtype called primary open angle glaucoma. That's the abbreviation stands for. No need to remember the full name. That's not relevant. Relevant is something else that is coming. Right? Why I chose to do POAG? Because POAG is the largest component. You can see is 50% of the patients have that subtype. And number two, that's the silent version. The other version associated with redness, pain of the eye and stuff like that. So they are detected early. Right? This one is, it can affect you from 10 years of your age to until 70, 80 years of your age. I'm very sure, I'm not going to ask, and you don't have to tell me, but I'm very sure in this audience there are people who knows at least one family member who has glaucoma. Right? It never happened that I did not meet one audience that when that's not true. It's that common. Right? And most of them are POAG subtype. Right? That's the painless version. That's the one gets long time to detect. Right? So that's what I'll be talking about. From a genetic point of view, and again as an example disease, because this is applicable to all other complex diseases that we know about, I'm assuming you guys understand monogenic disorder, multifactorial disorder, right? So this is a multifactorial disorder from a genetic point of view, and as most other multifactorial disorders, it has a rare component which works like a monogenic disorder. Right? It is a smaller subset, let's say 20% of all POAG are Mendelian classical genetics, right? And usually they are autosomal dominant form, right? Usually. There are exceptions, but usually. So the part on the left are the genes which can get mutated for the Mendelian form of the disease, right? Part on the right, which is the multifactorial type, that's about 80 genes. As you know, there are GWAS studies, and we have done all that, but I'm not going to talk about that today. Right? All genes put together explains about 7 to 8% of the genetic causes of glaucoma. And that is usual. Just to give you a context, you must know about BRCA1 and BRCA2 gene for breast cancer, right? Any guesses? BRCA1, BRCA2 combined, how much percentage of breast cancer it explains? 70 is 5%, right? So that's typical. The reason is that single gene disorders will often be more aggressive disease. They will happen early in life, so they will be selected against. They will affect your reproductive fitness, so those will always remain rare in frequency, right? So by design, by definition, something that works particularly as a dominant disease, works in a classical Mendelian fashion because there will be highly penetrant mutations, right? They will always be rare. They can never be common because there will be selection pressure on them. Multifactorials will be common because multifactorials are multifactorial, so any one factor is not as penetrant, right? That's why. The gene is on the red is called myocilin. Why myocilin is put on the red? Because all genes put together, it explains 7-8%. Myocilin alone explains 5%. So that's the most important gene if you're looking at the monogenic component of the disease. So today, we're going to talk about the monogenic component of the disease, particularly the myocilin gene, and what happened for a particular family. This is one family story. Little bit about the gene. Gene is very tiny, small, only 3 exons. And 90% of all the mutations known are in exon 3. Right? I don't want to go to the detail of biochemistry, but there are reasons for it. We have published long back that there are 2 blocks of nucleotide stretches on exon 3. One is block of 11, one is block of 9. And that 20 bases are mutational hotspots. These 20 bases account for 30% of all the mutations in this gene. Right? So we found that out. And the mutation that this family has is in one of the hotspots. Right? This is one of the hotspots mutation. Before I get to the family and get to the actual disease, actual findings, I'll give you a broader perspective, like zoom out, before you zoom in to the family. Whatever you are studying or doing your research on, it always has one single objective. Can you explain the phenotype looking at a genotype? Right? If you're talking about DNA. If I go beyond DNA, then can you look at a molecular signature that explains the phenotype? Right? That's, all of us are trying to do that. Right? Can I explain a disease by looking at some molecule, DNA, RNA, or protein? Right? And when DNA variation alone explains a phenotype, we technically call it a highly penetrating mutation. Why it's highly penetrating mutation? Because these are layers of information. The information flows inside out. Right? DNA information comes to RNA, information comes to protein. Everything put together and with interaction with the environment, phenotype is expressed. Right? So when I say classical autosomal dominant disease, that means what? That means looking at a mutation, I can tell whether this person will have the disease or not. Right? What does it mean? That all the other layers doesn't matter. Right? So that technically we call a highly penetrating mutations. Right? But think of it, so G2P stands for genotype to phenotype. Right? And G2P is always a very complex interplay of many information layers. Right? Today's story is going to be the simplest G2P. What is the simplest G2P? When a DNA information alone can explain that because of highly penetration and because that's where you can take an action. Right? Just to kind of give the context, I work on all the layers. I work on RNA, non-coding RNA, everything. But today I chose to talk about the simplest form of genetics, but a different way of handling it. Okay, so now getting into the zoomed in version, very boring slide to look at, and this is by design. So this is the sequence that we came across when we first got to interact with this family, and this sequence comes from exon 3 of myosin gene, right? And the highlighted C was a T in one particular patient. Right? And that patient, and why did we look at that patient? And why are you showing the slide in this boring way? Because to impress upon you, this is, you know what it is, you know how to read these things, but to impress upon you that the things you are doing in the lab, if you are a PhD student and getting frustrated with your progress or lack of it, you are trying to do something incredibly hard, incredibly difficult, and incredibly sophisticated. Right? You may not be doing this, it will be something else, but molecular biology is very complex. So I show this in a boring way to impress on that unless you don't know how to read this language, you would not even notice this change and not know what that means. Right? That's not trivial, the things we all trying to do. Right? Anyway, coming back to the story. So the index patient was brought to us by the clinician, and this is how her eye looks like. If you have never seen this kind of a picture, you would not know what to look for. So this is called fundus photography, fundus picture. And fundus, so this is the 4 different views of 2 eyes, left eye and right eye. And it gives you an idea of if the vasculature is normal in the eye, right? And it also tells you this bright spot is at the back of the eye where the optic nerve as a bundle goes to the brain. Right? And you may not be able to see it, but there is a yellowish layer here on the side. Right? The yellow layer for a healthy eye will be thicker and all round. Yellow layer indicates that the thickness, the amount of nerve cells, the neuronal cells that carries the signal back to the brain. What we are seeing here, that most of the neurons are almost gone. All the neurons are dead. So it's a neurodegenerative condition. Right? Neuronal cells that takes the information to the brain, ganglion cell it's called, those are the cells, they die. Right? So in glaucoma, another thing I should have mentioned in the beginning, think of a camera, nowadays digital camera, which doesn't have a memory card. Right? Everything else is fine. Right? In back days, the film camera-based cameras, everything is fine, lens is beautiful and all that, there's no film. Right? So that kind of thing happens. The eye is perfectly fine. Everything works. It processes the information, but nothing is there to carry the information to the brain. So it doesn't make sense. Right? That's what happens in glaucoma, and that we can tell from this kind of an image. Right? This yellow bit here, if you can see, is there a pointer? This yellow bit. I thought the mouse was working, sorry. Mouse is not working, my mistake, sorry about it. Thank you for pointing it out. So yeah, that's the bit. This yellow bit, this should be more round and uniform across the board.
This is string.
Sorry?
This is string form and easier.
This is the ring form, yes. It's called optic disc cupping. It's cup to disc ratio. The ratio decreases as the neurons are lost, right? You remember that peripheral vision loss video, right? As the cupping gets thinner, you lose your peripheral vision. So that's the connection between the two. So the mutation that I showed you, a C2T change, that happens to change an amino acid from proline to leucine at the 370th codon in the myosin gene. This mutation, when we discovered it, was already reported by other parts in the world, and everywhere it was reported, it was always associated with a very aggressive form of the disease. In other words, it was a well-reported, highly penetrant mutation. Okay? So the good thing was that, the bad thing was there was no novelty in the mutation. The good thing was that we didn't have to prove that it was a mutation. It was already established. What we did, we first of all confirmed it. So this is the sequencing data. If you know how to look at them, then you've already spotted the heterozygous peak. And so we confirmed that this mutation is there. And we also developed an RFLP assay, restriction fragment based assay, where you can do it much cheaper and much quicker. right? So this assay today, if we do it in RFLP way, it will cost you 75 rupees per DNA. So it's that cheap, right? So that was my intention, making it affordable, making it a diagnostic that is very easily done, right? You can just look at the band position and tell who is positive, who is negative. So this was 20 years ago, right? And this was the family. That's the index patient. I'm guessing again you know how to read a pedigree, right? That's the index patient. And through the index patient, we learned that there is a wider family and many people in the family were blind, right? This person was also legally blind, right? So we got into a situation where we were invited to their village. It is in rural West Bengal that I'm talking about. And we went there and we collected samples. And sure enough, as a classical autosomal dominant disease, everybody who had the disease had the mutation. Everybody who didn't have the mutation didn't have the disease. So it was 100% match in that way. The text in yellow, no GC involved, means at that time the concept of genetic counseling did not exist, right? There was no genetic counselor involved, right? So I had to do the counseling job without knowing that it is called a counseling job. Basically, I had to educate them, convince them that what is the pros and cons of getting into that. And remember, this family largely was illiterate and they were blind, majority of them. So it was not an easy task, but a lot of fun to go through that challenge. Now I'll switch a little bit from the data. When I went there, this is how they live. This is the actual house. Even now they live like that, right? And then because we figured out the mutation in the family, and you guys will understand it very well, that meant that somebody who is a 5-year-old kid, for example, right, we could tell whether they will have the disease or not when they become older, right? They had no symptoms, they're absolutely normal. But we could know and we could tell that this boy or this girl, when they turn 14 or 15 or 16, they will have glaucoma and they'll be blind by the time they're 18. So in this family, because it's a very aggressive mutation, anybody who had the mutation was fine until they were 10 years old. From 11, 12, they start losing their peripheral vision. How they know? They have to turn, look at the side, and by the time they're 18, they're blind, right? So imagine now, think now like a human, not like a scientist, right? Think like a poor family or anyway struggling, right? They have a kid which they're hoping they will grow up, they're studying in school, they will get a job and they will help financially. They are fine until 12 years old, happy child playing around. By the time 18, in 5 years' time, they're blind, their life stops. And it's like a time bomb because they don't know who will have it, who will not have it. Doctors can only detect it when they realize they have a problem and they visit the clinic. By that time, 70% of the vision is gone, right? And that's what started bothering me. What was bothering me? That I know who will be blind. I can tell the doctor who will be blind. Why doctor cannot do something about it? This kept bugging me. And the problem is, for very good reasons, for data objectivity, for data privacy, for patient privacy, you cannot show the patient. You cannot bring the patient to a lecture like this, right? Because then they will be harassed, right? So I had to always talk with the data, with objective data, and that never get beyond the conference rooms, right? And I wanted somehow to help those people. So back in 2023, after 20 years of struggling with this idea, I got a team together and I decided to tell their story through a film. I decided to make a film by hiring actors who will play the part of the patient. So what happens? I get rid of the privacy problem, right? I'm not showing the patients. I'm keeping the story real, but not showing the people, not revealing their identity. So notice, I have not told you where exactly they live. I did not tell you the name of the village or name of the district, right? And I'm not going to share their name, right? So I made a film. And I'll not show you the film today. It's available on YouTube to watch. I'll show you a 2-minute trailer now, after this slide. And then I'll talk about what the film has done, which I could not achieve any other way for 20 years. So this film is called The Vision of the Blind Lady. And this is funded by my university. The film making is funded by my university, but it was made in West Bengal, made in Kolkata. So we actually shot, all the scenes you will see is shot in that village in front of their house. So we kept it very, very authentic. So we got all the crew. So the filmmakers are from West Bengal themselves. And so this is the QR code to go to the link of the film. And the below QR code is, I gave a TED talk recently last year on this. And that TED talk is also available to watch for free on the YouTube. And so that's available in these two QR codes. Right? I forgot to say that language is in Bengali, but hopefully you saw the subtitles. And we purposely kept it in Bengali to keep it authentic, because we are telling the story of a family who is from Bengal, so that to keep that authenticity. Now, this film has started to change things, which I was very surprised about. So this film was released in 2023, 2 years ago, at the same time, in May. And when this happened, clinicians came forward. So these two, she is a genetic counselor and he is a clinician, who came forward and joined the movement. And they started to do things, started to move the ethical boundary, right? And so now what we are doing for this family, we are doing a pre-test counseling. So we are educating the family. I am not doing it anymore. A professional genetic counselor is doing it. If they are found, they are agreeing to the consultation, then the doctor is getting their eyes checked to make sure whether they already have the disease or not yet. And at that point, a sample is taken and the mutation is screened, right? Which is happening in NIBMG West Bengal and in Calcutta University Department of Genetics by these two colleagues of mine. And if the genetics confirms they have the mutation, then they are going to a post-test counseling, which is they are being told what to expect, and then doing surgery if required, if the disease is so aggressive. So there is now a management strategy, right? And that has changed things. How it has changed things? I did not tell you one particular aspect of the family. This is the same pedigree, a much smaller way I'm showing you. You know how to read a pedigree, but you probably don't know what this symbol means. Oh, sorry, again, I forgot. You probably don't know what this symbol means. I'm surprised if anybody knows, because I made it up. There's no nomenclature for this symbol. So that symbol, we created that symbol. That symbol means they were pre-symptomatic. They had the mutation, but they were young enough that they are not showing any symptom. Right? And this was 20 years ago. And this was troubled. This is what troubled me. So I was able to predict then, back in 2002, that these 3 kids will become blind. Nobody did anything. Nobody believed me. The family didn't believe me. The doctors didn't believe me. And nobody did anything. They waited until they lost 70% of the vision. And I now go back to the village every time I'm around, and yes, they are blind, which is not a surprise to any people who understand genetics, but they are surprised. So the bad thing is they gone blind, but the good thing is now they believe me, because they can see whatever I predicted happened. Right? So they think I'm some kind of magician or something like that. So what has changed? So these 3 people I could not save, but now they have children. Right? So now they have children, and completely out of coincidence, these 3 kids are also at the exact same stage which their parents were 20 years ago. Meaning they have the mutation, they not yet have the disease. Right? What is different? Now we have the film. Right? Now we have the film. So what the film has done, because of the film, the clinician has operated all these 3 kids. You have to wait until some symptoms is shown. That's a medical guidance, which is I agree with. But you don't have to wait when the vision is lost. So now the doctor is waiting only when they show very early signs of the disease. What is an early sign? There is something called intraocular pressure. Pressure inside the eye increases before they start losing the neuronal cells. Right? So now these 3 kids have undergone surgery. The moment their IOP starts to go up. Right? And this is one example. So this is one of the patients. Of course, not presented in an identifiable way. And this is just an example, and this is true for all the three. So all the three has been operated 1 year ago, last year. They've been followed up every month, and their disease has not progressed. So we have been able to save blindness for 3 kids. Not because of the science alone, because of the film. Science was there 20 years ago. There's nothing new scientifically has been done. Nothing new has been discovered, right? The people who are not convinced looking at the objective data, now they are convinced looking at the story, looking at the human side of the story. And that is what, if you forget everything after the moment you step out, that's fine, but only remember that, right? That what changes when you tell the human story. I'll tell you that, what changes and how does it change, right? So the 3 cases of blindness saved we have achieved already, and now I'm in Delhi because now I want to make it into a national policy. So I'm having a meeting with policymakers on Monday in Delhi. So that's the reason I'm here, primary reason. So this has gone beyond now one film, one story. And that's the power of storytelling, right? Which objective science alone do not have. Now, why we need this? Why is it needed? This is a very complex side. Don't bother reading it. The reference is there. Just look at how busy the circle is on the left compared to on the right. Right? The busyness shows the number of rounds of clinic visits and interventions you need to do without an early diagnosis, using genetics or otherwise, without early diagnosis. And the right, the less crowded version on the right shows if you do an early diagnosis, somehow, I'm saying using genetics, but somehow, how many less visits you have to do and how much less expensive it becomes for the society as a whole, right? This is done in US. So just to give you an idea of the numbers, anybody with one rare disease that happens early in life, to manage that disease in a lifelong span is cost $220,000 US for one person. The cost to society, that is, right? Medicine, doctors visit, time, lost opportunity, lost jobs, all things put together, that's the cost, right? And if you do early intervention for diseases that you can do something about it, like here we are showing that we can do an early surgery and you can actually save blindness. That means these kids will need management, will need eye drops, but they can get a job. They'll be able to see, they can complete their studies, right? Which means you are reducing the money loss, right? So that 220,000 comes down to something like $40,000-$50,000 for one person. That's the saving, cost saving. So that's why you do it. So I would like to call it was not a term coined by me, many people call it. I would like to call it actionable genomics, where you can take an action, right? Which is different from predictive genomics. When you are sequencing a genome or doing a GWAS study for a multifactorial disease, that's a predictive genomics, right? We do not need, for actionable genomics, we do not need the sophisticated toys. We don't need to sequence every genome that we see, right? This particular case is solved by a 75 rupees RFLP test. For this family, as long as it takes for generations to come, anybody from this family can know whether they have the mutation or not by paying less than 100 rupees. You don't need high machines, a scientific lab, nothing, right? Anybody can do it. So this is what is said in bullet points, but that is another message I want you to remember, that genomics is a tool, right? Genetics, genomics are tools. Use the component of the tool depending on what is the purpose. If my purpose is doing it that can I save blindness, then I don't need to sequence the genome where I know the gene I need to sequence, right? So actionable genomics. And I keep saying that the scientific is nothing new for 20 years, but the way we communicated is new, and the storytelling changed. Why storytelling has changed things? Because when we subjectively hear things rather than objectively hear things, different parts of our brain gets engaged, right? Your feeling center and your logical center are different. Your feeling center, which is the back of your brain, hippocampus, amygdala, those regions, they don't understand logic, they don't understand language, right? When you are trying to understand the mutation and RFLP study, you are not feeling anything, right? You are engaging your frontal cortex that understands logic, do not understand emotion, right? I'm trying to bridge the two. Why I'm trying to bridge the two? I'm sure all of you would have a favorite story that you heard as a kid from your grandparents or a parent, right? You can, in a moment, you can close your eyes and go back to that feeling, right? I'm also sure you don't remember the paper you read last week. Why that is? Because the paper did not make you feel. So you don't remember if you are not emotionally charged, right? This is what I try to change. So I'm talking to an audience like this, showing very hard objective data, showing lots of publications. You're not feeling anything, right? I wanted people to feel. And that has changed things. And how that all makes sense? How should you think if it appeals to you? Because you have a longer future than me. Think of the story are nothing but data with a soul. So if you put soul into the data, if you put feeling to the logic, it becomes a story. So telling story doesn't mean you have to move away from very well done science. Science can be still very robust, very objective, but it can be, you can still make people feel the human side of it, right? And that's where I will end with. These are the people who did most of the work. So the clinician, the genetic counselor, the geneticist, this Professor Kunal Ray that you may or may not heard about is a very senior scientist. He was my PhD mentor, and this project started when I was a PhD student, and these two were my juniors, so in the same lab. So it's from that lineage, and primarily funded by my university here in India. IGIB and Premas are my partners in this mission that I have taken up. I kept these two people for the end. They are the filmmakers, okay? They have nothing to do with science, so if the story appeals to you, it's their credit, right? They took my information and converted it into an emotional narrative, right? So that's what I wanted to acknowledge, and thank you for listening, and I'm happy to take any questions. Yes?
Is there any progress in diagnosis along in its cure also, like people are researching over?
Cure for glaucoma? A lot of people are, but because it's the problem is death of differentiated neuronal cells, cure is almost impossible. Management is mostly by surgery and then eye drops. For very severe cases, they start with surgery, so this family for example, and then maintained by eye drops. For milder cases, they manage a little bit longer with eye drops and then they do the surgery. Surgery, basically the intraocular pressure that increases because there is a fluid build up inside the eye, and because there is a channel that blocks the fluid, right? That's the physiology of it. And the surgery is nothing but creating a bypass, so the fluid can come out to another way, so the pressure is reduced, and if the pressure is reduced, then there's less pressure on the neuronal cells, then they die slower. So that's the hope, right? That's the only management. There is no cure. This is a particular condition, again, because it's differentiated neuronal cells, you cannot cure it by CRISPR. You cannot do gene therapy, because how many neurons you will cure? This is not a stem cell based thing, right? So that will not happen unless the only thing is if you can grow like a ganglion cell in the lab and remove the ganglion cell, put it back. But it sounds like a science fiction even now. So there's no cure. There's a comorbidity. There's a lot of glaucoma patients have diabetes and a lot of diabetes patients have glaucoma. But I am not aware of any molecular connection. So nobody has shown that this gene causes both or these two genes are working together. That has, as far as I know, nobody has shown. But yes, there is a very strong comorbid connection between the two. Most of the connection is a diabetic person develops glaucoma, and that is usually because the sugar, the high blood sugar, makes the blood vessels inside the eye, because they are very thin, blood vessels corroded, basically the sugar, right? And that sometimes makes it difficult for the oxygen and the nutrition to reach to the ganglion cells. And so basically they become starved cells, and that's why they die. That's what I know, but molecularly, as far as I know, there's no connection, no. Yes, there are, but it changes from one subtype to the other. So primary open-angle glaucoma, the subtype I'm focusing on, that subtype, if you look at a proportion of patients, all patients, most of them are adult onset. So 40 onwards. Right? It's an old age disease, right? And that's why they are multifactorial. They are less aggressive, less penetrant mutations, right? This family and all the other families where this runs as a monogenic condition, happens much early, that happens in the teens, and they will be blind before they are able to reproduce. That's how basically evolution works. So evolution works that way, that the penetrating mutations the selection pressure will not like to let it get passed on. And that's how the frequencies are kept low by natural selection. So those will happen early on. The other subtype, the primary congenital glaucoma, as the name suggests, is congenital. So it happens at birth, very, very early. But that's very painful. That is immediately detected.
So if someone is affected like around age of 20 to 25, let's say, so is it like chances like it will be cured?
No. No. So let me give you the blunt answer. No. But the earlier it is, the better managed it can be. And because it is irreversible, the earlier it is caught, the less vision lost, the more longer vision will be extended in their life. So this, again, coming back to this family, for example, even in this family, if you leave it unchecked, by the time somebody is 17, they will lose 70-80% of their vision. They will be legally blind. They will never get a driving license. They will never be able to hold a job. They will have to go to a blind school. And blind school needs somebody who is blind at birth. So these people are not even allowed to enter blind school. So they are left nowhere. So then they are not even 25. They're 17, 18, their life stops.
Proper lifestyle can be very
Sorry?
proper lifestyle.
Yes. So what you are talking about is a big part of our efforts now, advocacy, rehabilitation. That every blindness, we are very lucky that we are probably, hopefully able to save 3 children's sight, right? But most of the things will not be this lucky. So then we need to find a way to rehabilitate them, to train them even if they don't have a lot of sight left, so that they can have an employment, so they are financially independent. That's the whole point. Now, this family, they are living day-to-day basis. So maybe two members who can see are dragging on 25 members who cannot see. It's that kind of a situation. So that is we trying to solve.
Research was limited to West Bengal or like it's
Oh no, this research is globally done. So globally done.
like in that research thing, did you like notice a change with respect to India and the other nations? Like India is ahead of the other nations in that, or vice versa?
If I am successful in this mission, getting into the policy, then India will be ahead. Not now, not yet. Somebody has to believe, who can change policy, somebody like that has to believe me. That's not happened yet. But the research for glaucoma genetics is global, and for any genetic disease that you may know of, mutations are not country specific. Yes, you can have population specific allele frequencies, you can have private mutations clustered into a community, but most of the time, particularly the mutational hotspots, aggressive mutations, anywhere in the world, anybody has a mutation, they will show the same disease. That's usually the case, right? So this is the case here. So this mutation has been found in every population around the world, has been studied, and every time it is a very, very aggressive mutation. My research for glaucoma genetics is global. I have done GWAS, I have done genome sequencing and all that, and doing all that, having the entire portfolio of experience, I think this is the best way. This is the best way, because doing that, I can publish high-flying papers, I can get a lot of badges, A lot of PhD thesis happens, but nothing goes back. Why nothing goes back? Not because people don't want to, but because we are not 100% sure. It's always a p-value. It's always a percentage, right? This is where we are 100% sure. It's very simple, intellectually not challenging, but the simple thing, if you can save lives, why not? Right? So that's
Of that differentiated neurons degenerate due to the intraocular pressure increase. So, will the intraocular pressure affect the degeneration of the neurons, or it is vice versa? How that is?
Usually the intraocular pressure is a pure mechanical stress. Think of the eye as a ball, right? The pressure happens in the front of the ball, but the middle size of the ball is hollow, like a rubber tennis ball or cricket ball is hollow. The back of the ball is the retina, and the last layer of retina is the ganglion cell. So when this part gets pressurized, where will the pressure go? Pressure goes inwards, because nothing in the middle directly comes onto retina. Right? So the pressure, the mechanical stress kills the cells. Right? But your question has a point. It's not other way around, but there is about 30% of glaucoma that shows the same ganglion cell death without any increased pressure. And we don't understand why. Part of it is the comorbidity with diabetes. So maybe because they have weaker vessels, maybe because the neural cells are not getting enough nutrition, enough oxygen, something. That's intellectually that's the most interesting part of it, because we understand very little of it. But if I'm thinking of translating work so that it goes back to public, then that's far, far away, because we still don't understand. It has to be something very simple if you are trying to translate. But that's the answer to your question.
Sir, are there any treatments that can delay or prevent the disease?
Yes, as I was explaining before, the usual methods are surgery and eye drops. That's the usual method. How much it will delay, it usually never stops, it only delays. How much it will delay depends on the person, depends on their overall habits, their lifestyle and all that. It also depends, for genetic causes, it also depends on where is the mutation and how important is that position in the gene for that mutation. So it will depend, so it varies from case to case. This particular case is a very aggressive mutation.
Any other question?
For the same person?
Yes.
Very good question. It's not, so it is when your cells are dividing, right? So I'm taking you as an example. Is that okay? Right? So your cells are dividing all the time, right? Dividing means the DNA is getting copied, right? So if you ask the question, does it not make errors? Yes, it does. There's a DNA repair mechanism which removes most of these errors, but not all. Some errors stay, right? But in the large scheme of things, you have a 3 billion base pair long genome. In the large scheme of things, the chance that in the same gene or within the few nucleotides, random mutations will keep happening in that particular cell type is extremely, extremely rare. So theoretically, yes, we do accumulate mutations as we age on top of what we inherited and on top of what is the de novo mutation. But most of the cases, the inherited conditions stay inherited purely because of the randomness statistics, because out of 3 billion nucleotides, each nucleotide is equally likely. So something to happen in the same position or very close by position is very, very good. Does that answer your question? So practically it would not matter.
And also with effective theoretical analysis like this mutation or this gene variation causing that this disease is not curable with a particular person.
Accumulating mutation usually will not matter for clinical intervention, because for example, if you think of genome sequencing, that's usually done once, assuming it doesn't change, because most of the time it doesn't change. Right? So it will be very very surprising if the same mutation gets demutated completely by a random mutation. And for glaucoma in particular, because the problems is the death of neuronal cells which do not divide, so the problem doesn't exist.
The lecture was very engaging, insightful, and thought-provoking. Let's have a huge round of applause for Sir. Now I request Professor Yasha Hasija to felicitate Professor Arijit Mukhopadhyay.
On behalf of Department of Biotechnology, I would like to express gratitude to all those who contributed to this event. And I'm thankful to our Honorable Vice Chancellor, Professor Prateek Sharma, whose inspiring words always promote us to do our best. Thanks are due to our head, Department of Biotechnology, Professor Yasha, and my faculty colleagues in organizing the event. Special thanks to all the students who participated and asked so many questions. Thank you, everyone. Thank you. I think this story, it has been well taken by the students, and I was able to see that so many of you were searching for that particular video on your mobiles. So you can see that later also. Thank you, everyone.
I request everyone to please proceed for a group photograph.